Medulloblastoma, MB. It's the most common malignant brain tumor in kids. Just devastating.
Absolutely.
And for years, a huge chunk, almost 40% of cases, this group 4 or G4 Melody, it was just, well, a complete mystery, wasn't it?
It really was. We understood some of the other subtypes like WNT and SHH pretty well, knew their origins, but G4, it was frustratingly obscure, defined mostly by, what it wasn't.
Okay, so today we're doing a deep dive into some really groundbreaking sources. They seem to finally offer a definitive explanation for G4 Melby.
That's right.
And it traces the origin not just to, like, a single bad gene, but to something really specific and unique about how the human brain develops. A structural thing.
Exactly. A structural quirk, you could say.
So our mission today is to really unpack that, understand the precise developmental failure that makes us, humans, uniquely susceptible to this terrible childhood cancer.
It's a fascinating story where genetics, development, and even evolution all converge. It's quite remarkable, actually.
So set the stage for us a bit more. You mentioned the subtypes.
Right, so MB isn't one single disease. It's highly heterogeneous. We classify it into 4 main molecular groups: WNT, SHH group 3, and the group 4 we're focusing on.
And G3 and G4 are the most common.
They are. G4 makes up about 40% of patients, G3 around 20%. Together, that's the majority of cases, and sadly, also where most deaths occur.
And we knew where SHH came from, the granule cell lineage.
Correct, and WNT from the lower rhombic lip.
Hmm.
But G4, the thinking was maybe the unipolar brush cell lineage, or UBCs, but it wasn't clear. Possibly an earlier progenitor stage.
Okay.
And what's really emerged, what's so fascinating here, is that the deeper we look into the timeline of human embryonic development, the less G4 Melody looks like a typical cancer that just pops up later from random mutations. It looks much more like a problem of arrested development.
Arrested development, like the cells get stuck.
Precisely. Frozen in time at a very specific, highly proliferative and vulnerable progenitor stage. Like they never grew up.
Okay, so if it's developmental, we need to start at the beginning. You mentioned the rhombic lip, the RL. This is where all MBs start.
That's the origin point, yes, the rhombic lip.
For those of us whose neuroanatomy is a bit rusty, what exactly is the rhombic lip? Why is this specific brain factory so crucial here?
Well, think of the RL as the primary construction zone in the developing hindbrain. It's the progenitor zone, the factory, that produces most of the neurons that will eventually form your cerebellum and some other nearby structures.
The cerebellum, balance, coordination, that part of the brain?
Exactly. And all medulloblastomas, regardless of the subtype, arise from cells originating in this RL factory.
Okay, got it. RL equals neuron factory. Now here's the twist, right? The human anomaly.
Yes. This is where the story gets specific to us. The research highlights a major difference when you compare human development to, say, mice or even macaques.
What happens?
Around 11 weeks after conception, so pretty early, about 2.5 months into pregnancy, the human RL undergoes this really dramatic structural expansion, and it physically splits.
It splits. The whole structure divides. Why would ours do that, and not a mouse's or a monkey's? That seems significant.
It is significant. It seems to be an evolutionary adaptation tied to the massive expansion and complexity of the human cerebellum compared to other mammals. Our cerebellum is just bigger and more complex.
So evolution gave us a bigger cerebellum, but maybe introduced a new structural complexity during development.
That seems to be the case. The RL divides into 2 distinct zones, both molecularly and structurally. There's the RLVZ, the ventricular zone, which mainly contains the earliest stem cells.
Okay, the stem cell reservoir.
And then separated from it by a network of blood vessels of vascular plexus is the RLSVZ, the subventricular zone. Let's call this the proliferative zone to make it easier.
The proliferative zone. Okay, I like that. Easier to remember.
And this zone is absolutely packed with progenitor cells that are actively dividing, proliferating.
So this split creates a whole new neighborhood, the proliferative zone, bustling with dividing cells, a human-specific neighborhood.
Exactly. A new microenvironment unique to human development at this stage.
Which specific cells are living in this proliferative zone? And why are they the ones susceptible to becoming G4 maybe?
Good question. This proliferative zone is home to a specific population of progenitor cells. They are precursors to unipolar brush cells, UBCs, and they express certain markers, like EOMEs and crucially Ki67, which shows they're actively dividing.
EOMEs plus Ki67 plus UBC progenitors.
Got it. And here's the key finding from the sources. This specific pool of cells is spatiotemporally expanded in humans.
Spatiotemporally expanded. Okay, break that down. Sounds like science. jargon.
It just means two things, really. One, these cells occupy a larger area of tissue in the developing human brain, the spatio part.
Okay, more real estate.
And two, they persist for a much longer time during gestation compared to other species like mice, the temporal part.
Yeah.
They hang around longer.
Ah, I see. So in humans, this population of potentially vulnerable, rapidly dividing cells is both bigger and exists for a longer window of time during development. That makes intuitive sense. If you have a vulnerable stage, and in humans that stage is stretched out over more space and more time.
Well, that inherently increases the statistical odds that something could go wrong, right? There's a mutation happening.
That's precisely the implication. It's like you said earlier, leaving a busy construction site open for way longer than planned. It increases the chance of accidents. This seems to be the evolutionary trade-off. We get this amazing, complex cerebellum.
But the cost might be this expanded developmental window that makes us more prone to G3 and G4 melum.
That appears to be the cost. And critically, when the researchers looked at the actual G4 myeloid tumor cells from patients,
Yeah. What do they find?
they mapped their gene expression profiles, their transcriptional signatures. And these cancer cells looked almost identical to those specific EOMEs plus Ki67 plus progenitor cells found in the human proliferative zone.
Wow. So the cancer cells are essentially cellular snapshots, frozen copies of this unique human developmental stage.
That's a great way to put it. Cellular snapshots stuck in developmental time. Okay, let's take a quick pause here for our first mini recap. This is fascinating. So just to bullet point for everyone listening.
A good idea.
First, medulloblastoma, or MB, is a cancer that originates in the rhombic lip, the brain's neuron factory.
Correct.
Second, uniquely in humans, around 11 weeks into development, this rhombic lip physically splits, creating a new area called the RLSVZ, or what we're calling the proliferative zone.
Yes, a human-specific feature.
Third, this proliferative zone is filled with a specific type of rapidly dividing progenitor cell, the EOME plus Ki67 plus UBC precursor. And this cell population is much larger and persists for much longer in humans compared to other animals.
That expanded window is key.
And finally, the G4 MIB cancer cells look transcriptionally just like these specific human progenitor cells. They're stuck at that stage.
Spot on.
Okay, so here's a question for you, the listener, to ponder. Does simply having a wider window of development, like this longer persistence of the proliferative zone in humans, inherently increase the opportunity for something like cancer to arise? An evolutionary cost of complexity. Something to think about. All right, so we've established the where, this unique human proliferative zone, and the who, these specific progenitor cells. The next big question is why? What's the molecular trigger? What goes wrong inside these cells to freeze them in that immature proliferative state?
Right. And this leads us directly to the core molecular mechanism identified in these studies. It turns out G4 MIB isn't caused by, dozens of random unrelated mutations.
No.
The genetic problems, the lesions, they consistently converge on one critical piece of cellular machinery. The core binding factor alpha complex. Let's call it the CBFA complex.
The CBFA complex sounds important, like a master controller.
You can think of it that way.
Yeah.
Or maybe like the engine that drives differentiation, the maturation process.
Okay, the differentiation engine. What's its normal job?
Its job is crucial. It's meant to push these progenitor cells out of that proliferative state, out of the proliferative zone, and guide them forward to become their final, mature, non-dividing neuronal form, like a fully formed unipolar brush cell or a granule neuron.
So it tells the cell, okay, stop dividing, time to grow up and get your specific job done.
Exactly. It initiates that maturation program. The complex itself involves several key proteins, especially adapter proteins called CBFA2T2 and CBFA2T3. These guys work together, recruiting other molecules, epigenetic modifiers, transcription factors to the DNA. They're the chemical signals that say, mature now.
And in G4 MIB, this engine is broken.
Fundamentally broken. And what's really telling is how it's broken. The mutations or alterations that hit this pathway are almost always mutually exclusive.
Meaning?
Meaning if a patient's tumor has one specific type of mutation disabling the CBFA complex, they typically don't have another type of mutation also hitting it. It strongly suggests that any one of these hits is sufficient to cause the problem. All roads lead to the same functional breakdown.
Okay, so what kind of hits are we talking about? How does the engine break?
The sources detail several ways this happens, all converging on the CBFA complex. One common way is through loss-of-function mutations directly in the gene for that adapter protein, CBFA2T2. These mutations essentially cripple the protein so it can't do its job.
Okay, a direct hit on one of the engine parts.
Right. But perhaps even more interestingly, We very frequently see large chunks of DNA missing focal deletions on human chromosome 16, specifically a region called 16q24.
A whole section of a chromosome just gone. Why is that location important?
It's incredibly important because that specific chunk of chromosome 16 happens to contain the gene for the other key adapter protein, CBFA2T3. So deleting this region wipes out or significantly reduces CBFA2T3.
Ah, so it takes out another critical engine part.
Yes, but it gets even more interesting. That same deleted region on chromosome 16 also contains several other genes that have been implicated as G4-MeRB drivers or tumor suppressors, like FANCA and ZFHX3.
Wait a minute. You're saying the way our own human genome is organized, the physical layout of genes on chromosome 16 actually makes us more susceptible to G4-MeLiBa because multiple important good guy genes are all clustered together, making them vulnerable to being wiped out by a single deletion event.
That's precisely the argument. This clustering helps explain a major characteristic of G4-MeLiBa. It's overwhelmingly driven by these large chromosomal deletions, what we call copy number aberrations or CNAs, rather than small, single-point mutations or SNVs.
Because one big deletion event during cell division can knock out multiple key suppressor genes at once. A bigger bang for the buck, genetically speaking, for the potential cancer cell.
Exactly. It's a catastrophic event that gives the cell a huge advantage by simultaneously crippling the differentiation engine, CBFA-TT3, and potentially other safeguards, like MNC ZFHX3. It really underscores how G4-MeLiBa seems tied to the structural vulnerabilities of the human genome itself, especially during the rapid cell division of embryonic development.
So the result is the differentiation engine, the CBFA complex, completely fails. The cell can't mature.
Correct. The accelerator pedal is broken or ripped out entirely.
Okay, so the CBFA complex is the accelerator, the engine driving maturity, and it's broken in G4-MeLiBa. But you mentioned earlier this idea of cells being frozen or stuck. Is there also maybe a break? Something actively holding these cells back in their primitive state besides just the accelerator being broken?
Yes, there is. And that brings us to another key player, a protein called OTX2.
OTX2. Okay.
Remember, we said G4-MeLiBa cells are stalled, transcriptionally similar to those proliferative zone progenitors, but unable to mature. Well, OTX2 is a transcription factor, a gene-regulating protein that is very highly active back in the early rhombic lip and in that proliferative zone.
So it's active early in development.
Very active early on, and significantly, OTX2 is often found to be amplified, meaning there are extra copies of its gene, or it's just generally overexpressed in both group 3 and group 4 medulloblastoma.
Okay, so it's an early developmental gene. It's overactive in the cancer. Does it act as a restraint? A brake on maturation?
That's exactly what the evidence points to. OTX2 seems to function as a powerful developmental brake. Its job, early on, is to maintain that primitive rhombic lip identity to keep the cells in that progenitor state.
And how does it do that? How does it put the brakes on?
The sources show a direct mechanistic link. OTX2 appears to actively inhibit the CBFA complex, the accelerator we were just talking about.
It inhibits the accelerator. How?
It seems to do this, at least in part, by directly binding to the promoter region of the CBFA2T2 gene. The promoter is like the on switch for a gene. By binding there, OTX2 likely prevents the cell from even making the CBFA2T2 protein, effectively stopping the differentiation engine before it can even start.
Wow. So OTX2 clamps down on the accelerator production line.
That's a good analogy. It keeps the accelerator pedal from even being installed.
Okay, this sets up a really elegant experimental test, doesn't it? If OTX2 is the brake, and it works by suppressing the CBFA accelerator, then if you could somehow remove OTX2 or turn it off...
Release the brake.
Yeah. Release the brake, then the CBFA complex should turn on, and the cell, even a cancer cell, should be forced to mature, right? Did they try that?
They did. And this was really a breakthrough moment described in the research. Scientists used techniques like OTX2 knockdown, basically, silencing the OTX2 gene in group 3 MeLiBa cell lines. Remember, G3 also resembles these early progenitors and often has high OTX2.
Okay, so they shut off the brake in the cancer cells. What happened?
The results were striking. Almost immediately, the expression levels of those key accelerator proteins, CBFA2T2 and CBFA2T3, shot way up.
The engine turned on.
The differentiation machinery roared to life, now freed from OTX2's repression. And the amazing part, the cancer cells started to spontaneously differentiate.
They started maturing on their own.
Yes, orderly differentiation, moving along a pathway that closely mirrored normal cerebellar development. They started looking less like primitive progenitors and more like maturing granule cells and other neuronal types. The malignancy was, in a sense, being reversed by forcing maturation.
That is actually profound. Okay, let's do another quick recap because this ties everything together beautifully for you listening.
It really clarifies the core problem.
Recap point 2, the stalled state. First, G4-MIMA happens because those progenitor cells in the proliferative zone get stalled, stuck in developmental time.
They fail to mature.
Second, this stall happens because of one of two main reasons, which are usually mutually exclusive. Either the differentiation accelerator, the CBFA complex, is broken, often by gene deletions like on chromosome 16.
Right, loss of CBFA-ATT2 or CBFA-ATT3.
Or the developmental break, the OTX2 protein, is stuck in the on position, often due to gene amplification, actively suppressing the CBFA accelerator.
Exactly. OTX2 binds the CBFA-FE22 promoter, acting as a transcriptional brake.
And third, the clincher experiment. Artificially releasing the OTX2 brake in cancer cells forces the CBFA accelerator to turn on, causing the cancer cells to spontaneously start maturing and differentiating like normal cells.
It confirms the whole model beautifully. Broken accelerator or our stuck brake leads to the same outcome, stalled progenitor cells accumulating.
So here's the next question for you to think about. If this cancer's core vulnerability is its reliance on staying young and immature, does that offer a completely different angle for treatment? Instead of just killing the cells, could we maybe force them to differentiate, to mature into harmlessness? That's a huge therapeutic implication, isn't it?
It absolutely is. It opens the door to thinking about differentiation therapies, not just cytotoxic ones.
Okay, so we have the cell type, the location in the developing brain, the molecular mechanisms, the broken accelerator, and the stuck brake. Now let's connect this back to the actual patients. Clinically, where do these G3 and G4 tumors show up in the brain? Is there a pattern?
There's a very distinct pattern. Unlike the SHH subtype, which often appears in the cerebellar hemispheres, the sides, Group 3 and Group 4 medulloblastomas almost always present right in the inferior midline of the cerebellum.
The midline. Specifically where?
In an area called the nodulus, or the flocculonodular lobe, right at the bottom, center part of the cerebellum.
Okay, why there? Does the developmental story we've been building explain that specific location?
Perfectly. It circles right back to the embryology. Remember that rhombic lip, the neuron factory, and our unique proliferative zone within it.
Yeah, the RLSVZ.
Around 14 weeks post-conception, so a bit later in development, that whole progenitor zone structure, including the proliferative zone, actually internalizes. It folds inward and becomes incorporated into the developing structure that will form the nodulus.
Ah, so the birthplace of these cells literally gets tucked into the very spot where the tumors later appear.
Precisely. And this region, the nodulus formed from the remnants of the RL, is actually supposed to largely regress and disappear around the time of birth.
So the G4 tumor location is essentially the anatomical graveyard where these susceptible progenitor cells were supposed to vanish but instead got stuck because they failed to differentiate.
That's the model. They get trapped in the location where the RL makes its last stand, so to speak.
And this idea that leftover embryonic cells in that area could be the source of MB isn't actually brand new, is it?
Not at all. This research actually revives a hypothesis that's about 80 years old. Pathologists back in the 1940s observed these small clusters of unusual, immature-looking cells. They call them atypical cell rests or cerebellar heterotopias, specifically in the nodulus of infants and young children.
Heterotopia is like tissue in the wrong place or that shouldn't be there.
Exactly. And they speculated way back then that these might be the precursors to medulloblastoma. But the idea kind of faded because the molecular link, the Y, was missing.
But now, with the understanding of the RLSVZ progenitors and the OTX2 CBFA pathway failure...
Now we have the link. These studies provide the molecular explanation for those old observations. The authors propose renaming these structures persistent rhombic lip lesions, or PeRLs.
Persistent rhombic lip. I like it. So a PeRL is essentially a little ball of those RLSVZ progenitor cells trapped in the nodulus after birth because their differentiation process failed. Either OTX2 was too high or the CBFA complex was broken.
That's the proposed model. These are the cells that failed to get the signal to mature and disappear. They are embryological remnants.
Are all PeRLs destined to become cancer?
Probably not. The thinking is that most PeRLs likely regress on their own eventually, or just sit there harmlessly, but they represent a pool of vulnerable, developmentally stalled cells.
A pre-malignant lesion.
Exactly, a pre-malignant lesion. And the hypothesis is that these PeRLs are then susceptible to secondary genetic hits, things like gaining an extra copy of chromosome 17, which is very common in G3, G4 millib, or other mutations that push them over the edge into full-blown cancer.
So the initial developmental failure, the OTX2 CBFA problem, creates the PeRL, the susceptible population. Then a later genetic hit provides the final trigger for malignancy.
That's the two-hit idea gaining strength from this research. The developmental context creates the fertile ground. Subsequent genetic instability ignites the fire.
This is really quite a comprehensive picture. We've journeyed from G-form elite being this big unknown, this statistical mystery,
A black box, really.
to understanding it as fundamentally an evolutionary cost associated with our uniquely large and complex human cerebellum.
It seems so. The specific way our rhombic lip expands and splits creates that vulnerable proliferative zone, the RLSVZ.
That provides the susceptible cells, the EMEs plus progenitors, and then the failure of that critical OTX2 CBFA regulatory axis, the brake and accelerator for maturation, prevents these cells from finishing their development.
Causing them to stall, persist as PeRLs, and become vulnerable to transforming into G3 or G4
It completely reframes the disease, doesn't it?
Yeah.
It's not just random chance mutation. It's a failure embedded in our specific developmental program, a failure at a crucial human-specific maturation checkpoint.
Precisely.
And the potential clinical implications here are huge. If PeRLs are the identifiable pre-malignant lesion,
Right, the source of the tumor.
the paper actually proposes the possibility of detecting them early, maybe through advanced MRI techniques in infants, or perhaps finding specific biochemical markers like oncofetal antigens, proteins usually only made during development in the blood or cerebrospinal fluid during the postnatal period.
The idea being, if you can spot the PeRL, perhaps you could monitor it or even intervene before it ever becomes a malignant tumor.
Which leads us right to the final and perhaps most challenging question this research leaves us with. It's something for you, the listener, to really mull over.
It's a critical, ethical, and clinical dilemma.
If we can detect these PeRLs in newborns or infants, these potential pre-malignant lesions, but we also know that most of them might just disappear on their own, might never become cancer. What level of certainty, what clinical standard of evidence would doctors need before they could justify intervening?
Intervention could mean years of stressful surveillance imaging for the child and family, or maybe even preemptive treatments with their own risks.
Exactly. How do you weigh the risk of a potential but not guaranteed deadly cancer years down the line against the risks and burdens of intervening on a lesion that might be harmless, especially in a developing child? That's the complex frontier this incredible developmental discovery now pushes us towards.
A profound question raised by understanding the deep origins of this disease.